Hello everybody, Happy New Year, and welcome to the last chapter that we're going to do before finals. So give it everything you got so that way you can boost that grade up before the end. Alright, we are starting the next unit, Unit 7, which is all about genetics. And genetics has a lot to do with what you look like compared to your parents and what types of traits you get and how likely it is you're going to pass something on to your offspring and so on. So in doing so, we're also going to learn a little bit about how our DNA gets mixed up when making eggs and sperm, and that is what accounts for the probability of passing something on. So we're going to learn a little bit about mitosis and meiosis along the way. Alright, so let's start with, before we get into how genetics works, we need to do a little bit of vocab. Now, genetics is the study of heredity. Heredity is how you pass things on to your kids or to your offspring. And so in order to study heredity, we need to know a little bit about genes. Now, we've learned in the DNA chapter that a gene is just a chunk of DNA, excuse me, a chunk of DNA that codes for some RNA, which codes for a sequence of amino acids, which codes for a protein. And proteins determine what we look like, how we act, and what traits that we have. So a gene is kind of the important key aspect here because genes are what we're manipulating. So a gene is just a chunk of DNA, a sequence of DNA that codes for a particular protein and therefore determines that particular trait. Now the genes are on DNA. And usually when we look at DNA, we look at chromosomes, this little guy right here. And as we learned before, that a chromosome is actually made up of 2 chromatids. And each chromatid, remember a chromatid is 1/2 of a chromosome, each chromatid contains a replicated strand of DNA. So when I look at a chromosome, I'm actually looking at the 2 individual chromatids. And the chromatids are supposed to be identical to each other. And on the chromosome and on the chromatid, you have what's called an allele. An allele is one of a different number of forms of a gene. What I mean by this is you get a piece of DNA from your mom and a piece of DNA from your dad that carry the same genes. Gene for hair color from mom, gene for hair color from dad. But you have 2 alleles. I could get the allele for mom's hair color, or I could get the allele for dad's hair color. So I've got 2 choices, 2 versions of every gene. So I have 2 versions, mom and dad's, of every single gene in my body. So each version we call an allele. So I go, well, which allele am I going to get? Am I going to get mom's allele or am I going to get dad's allele? Well, that depends on if their allele is dominant or recessive. A dominant allele, right here, dominant is going to be the form of the trait that shows. So if I have a dominant gene or dominant allele, that's the one I'm going to show, no matter what. Doesn't matter if I have 1 of them or 2 of them. If I get 2 dominants, 1 from mom, 1 from dad, or 1 dominant from, excuse me, cat. Hi. Wait, see if I can do the cat beard thing. No, okay. So if I get a dominant one or 2, doesn't matter, I'm going to show that trait. Okay, so like having earlobes that are free and not attached to the side of your head, that is a dominant trait. Now a recessive trait is one that only shows up if there's not a dominant one around. So a recessive trait will show if he's paired with another recessive. So if I get the recessive form that my ears are attached to the side from mom and the recessive form from dad, well then I'm going to show recessive earlobes. So it all depends. So I get 2 of everything. I get 2 alleles for every protein, 1 from mom, 1 from dad. If they're dominant and dominant, I will show the dominant trait. If they're dominant and recessive, I'll still show the dominant trait. But if it's recessive and recessive, then I'll show the recessive trait. Okay? And we'll get into some common things that we have that are dominant and recessive. Okay. Now, how did this all come to be? Well, it's all because of this guy named Mendel. Mendel is this Austrian monk who had a lot of free time because he was a monk and all. And so he decided to study pea plants. So over here you can see that pea plants come in lots of variety. They can come in purple or white flowers. The flowers can be somewhere along the branch or at the tip of the branch. The peas themselves can be yellow or green. They can be round or wrinkly. The pea pods can be inflated or constricted. They can be green or yellow. And the plant, I'm going to lift it up here, can be either really tall or really short. So he decided to see what controls these particular traits. Well, he didn't know anything about DNA and proteins and stuff like that. So he didn't know that proteins controlled the traits that he saw. So he wanted to do some experiments. So what he did is he bred these plants to be pure. So pure purple or pure white. And pure just means that they're kind of like purebred dogs. It just means that they have nothing else mixed in them. They're not blended with anything else. So a purebred German Shepherd should only have German Shepherd blood in him. If he has a mutt in him as well, well then he's not purebred. So a purebred purple flower only has purple in its blood, we'll say. So he bred these plants to be true, or true breeding, pure plants that would only produce identical plants. So a purple flowered plant would only make purple flowered babies. A German Shepherd dog, purebred, should only make German Shepherd babies. If he makes something else, he's not purebred. So he made all these plants that were pure and he decided to cross them. So his little experiment was to breed them with each other and to cross two plants of known traits to see what their kids would be. So he crossed a pure purple with a pure white and he wanted to see what he got. Well, if you cross a pure German Shepherd with a pure Dachshund, you're going to get a hybrid. You're going to get a mutt, okay? Well, mutts in science talk, we call that a hybrid. So that's just a blend. So a hybrid is a plant that comes from two different traits, or a dog that comes from two different purebred parents. And what he saw is in the results, they all came out one of the two traits. So like he mixed purple and white, they all came out purple. He mixed axial and terminal, they all came out axial. He mixed yellow and green, they all came out yellow. And he thought this was really weird. Where did the other trait go? I don't know. Let's see. So before we get into the results, let's some more words here. Okay, the P generation, P stands for parent or pure. So he had to have some pure plants in order to start his little experiment. So the P generation, we call that the parent generation, and they were pure. So those are your purebreds. And when he crossed them, he got the first generation of kids. Well, we call that the F1, first generation. F stands for filial, which means son. And so F1, filial one generation, just means your first set of kids. So the P generation makes the F1 kids, and these are the offspring. These are the hybrids. So the purple and the white flowers made all purple. Well, those were the F1 generation, and they were all purple. But then he thought, well, this is kind of weird. Let me take the kids, mate them with each other, and see what we get. So he took the purple kids and crossed them with each other to see what would happen. And when it happened, when he got the pea plants, what he ended up was with about 75% of the kids had purple flowers and 25% of the kids had white flowers. And he said, oh, my white came back. That's cool. And he did it with all of them. He did axial and terminal. 100% of them came out axial. Crossed the kids. What happened? 75% came out axial, 25% came out terminal. And he said this is really cool. So his F2 generation, which are the kids of the F1 or the grandkids of the P, they produced a 75 to 25% ratio. And he said, well, that's really cool. I wonder why that is. So now at the time, again, he didn't know about DNA and alleles and all that kind of stuff. So he had to figure out what was going on. So all he could do was say, okay, this little cartoon goes with this little description here. So he took a tall plant and a short plant, and he crossed them, and they all became tall. Then he crossed two tall plants, and he got 75% tall, 25% short. Okay, so what happened? One of the traits disappeared in the F1, but reappeared in the F2 generation in a ratio of 3 to 1, or 75% to 25%. Okay, so why? Why did the one trait disappear and then reappear? Well, that led him to come up with the whole idea between dominant and recessive traits. And this allowed him to come up with the conclusion that each of the traits on the plants the color, the pea pods, all that kind of stuff, was controlled by what he called a factor. He didn't know what a factor was. We know what a factor is now. It's a gene. It's a chunk of DNA. And that they come in two forms, two alleles. And so we called this the principle of dominance. That some of those factors or alleles were dominant and they showed up more often, and some of them are recessive and they showed up less often. He didn't know why, so he continued doing experiments and such, but he at least knew that of the two forms of each trait, one was dominant over the other. And when he bred two pures together, the dominant ones showed up. But when he crossed two hybrids together, he got mostly dominant and some of the recessive again. And for that day and age, that was a huge deal. So what did we get out of it? Well, actually not that slide yet. What did we get out of it? We learned now that there are dominant traits and there are recessive traits. They are controlled by segments of DNA, genes on DNA that control proteins. The proteins are what control what you physically see. The color, the pea pods, the height of the plant, and so on. Now, if you look on the plants, you can see that there's two letters. Now, Mendel didn't know any of this stuff, but we use letters to represent the alleles, whether they're dominant or recessive. We use a capital letter to represent dominant alleles. We use lowercase letters to represent recessive alleles. So what really happened is that he had a purebred tall and a purebred short. When he mixed them together, he got hybrids. So these are mutts, okay? And so these hybrids he mixed together, and when he did that, he got a pure back, he got two hybrids, and a pure recessive, okay? We use Punnett squares to figure that stuff out. We'll talk about that later. Okay, last slide for today. And this will lead us into being able to talk about these things. Okay, so how is it that I get some things from mom and some things from dad? Well, it has to do with this thing called the law of segregation. The law of segregation says that mom and dad, when they make eggs and sperm, that the chromosomes, they're going to split. And they're going to segregate. They're going to separate into various eggs and sperm. And how those eggs and sperm come back together will determine what trait the baby has. So the law of segregation says that during meiosis, which we will talk about in a little bit, the two alleles will separate independently of each other. They don't go together, they separate. And that will lead you to your probability or the likelihood that something is going to occur. So if I have a redhead and a blonde and they have babies and I want to know what is the probability that their kids are going to be redheaded, well, then I can do Punnett squares to help me figure that out. So probability is just how likely it is that something will occur. Now, I showed you the letters, the big T, the little t's. We call that the genotype of the individual. It's what does our DNA say my protein is going to be. So I have DNA that says my ears are going to hang free. So my genotype is dominant for that allele. My husband, he's got those ears that just go straight into the side of his head, and they're attached. So his genotype says that his allele has the recessive form. So our kids could either have the dominant or the recessive form, depending on whose allele that they get. So we call this our genotype. The genetic makeup of an organism determined by their DNA. We represent it with two letters. So you can see the genotypes of these little guys, big A to little a, big A to little a. And then here, the genotypes of the babies could be big A, little a, or little a, little a. So that's the genotype, always represented by two letters. And then we have the phenotype. What do we see? That's the physical appearance. So I remember pH phenotype, pH physical. So it's what do I see? My phenotype is I have brown hair. My genotype is big B, big B. My genotype might be, we'll use E for ears, big E, little e, but my phenotype is I have free ears. So my genotype is big H, little h. My phenotype is I have hazel eyes. So you see the difference? The genotype are the two letters that represent my DNA. The phenotype is what do I physically see? Now, it doesn't have to be seen with your eyes. Your blood type. Your genotype might be big A, big B. Your phenotype is you have type AB blood. Okay, so you may not physically see them, but it's what you express, what you show. I show type A blood. That's my phenotype. My genotype could be big A with what's called a little I, or big A, big A. We don't know. So, genotype letters, phenotype, what do we see? Okay, I hit 15 minutes, so I'm done. So tomorrow we're going to take a look at common traits and see which ones you guys have in common. All right, talk to you later. Bye-bye.